So, my name is Dr. Luke Isbell. I'm a group leader at SAiGENCI in the Cancer Epigenetics program, of which the two enigmatic speakers you'll hear from tonight also. And it is my great delight and honour to welcome you here tonight for a fantastic public lecture. So, on the behalf of Professor Chris Sweeney and the rest of the SAiGENCI team, I'd like to welcome our alumni, our donors, partners, and all of the members of the community who are here today. It is really, really special that you can make it, and we really appreciate you for coming. Also, for those people who are joining online, either interstate or overseas, truly, thank you for coming. I'd like to begin also with the acknowledgement to country. So we acknowledge and pay our respects to the Kaurna people, the traditional custodians of the ancestral lands upon which we gather. We acknowledge their deep feelings of attachment and the relationship of the Kaurna people to the country, and we respect and value their past, present, and ongoing connection to the land and their cultural beliefs. A few housekeeping issues before we start. Toilets, if you need them, are out of the building and to the left. It's well signed, I'm sure you'll find it. And mobile phones, if you can turn them on silent, that'd be fantastic. Check them now. It's going to be much more embarrassing when they go off during the talk. So thank you. A little bit of background about SAiGENCI. So the South Australian Immunogenomics Cancer Institute, SAiGENCI, is the newest cancer research institute in South Australia, and it's the University of Adelaide's flagship cancer research program, which is a collaboration between the Central Adelaide Local Health Network, CALAN, and our goal is very simply to have fewer people get cancer and to improve the lives of those who have. And it's a very exciting time for SAiGENCI, especially over the last few years, where we've been heavily driving recruitment. And we've been trying to get the best and brightest that we can access over the world, recruiting them to Adelaide. I myself am originally from Australia, and I've spent several years abroad in Switzerland before starting my own lab here in Adelaide, where I'm very lucky to find my new home. So, our annual public lecture is our way of thanking you, both the supporters and community members, because you bring fresh insights and a really grounding force for us with which to tailor our research. In a way, we really work for you, the public. It's what kind of guides us, improving health outcomes and that knowledge that allows us to deliver new therapeutics to the public. It really is a strong sense of connection that we feel with you, and so we really appreciate you for coming. Without you, we cannot do what we do, and so very grateful. The topic for tonight's talk is Decoding Cancer, How Epigenetics Can Solve the Cancer Riddle, and I'll leave it to our fantastic speakers to actually go into depth on that. But after the talk, you'll get the opportunity to ask a few questions, and if you're particularly shy, you can even come up to the speakers afterwards and ask some additional questions. So without further ado, I'll introduce our fantastic speakers for tonight. First is Professor Jose Polo, who's a Professor of Epigenetics and Director of the Adelaide Centre for Epigenetics, also known as ACE, and he's also the Program Leader for SAiGENCI's Cancer Epigenetics Research Program. And we're located within the Faculty of Health and Medical Sciences at the University of Adelaide. Jose started at the Harvard Stem Cell Institute before establishing his own research group in Melbourne at Monash University. He's a fantastic researcher who's done some really groundbreaking, pivotal work in epigenetics, embryogenesis, and continues to work on those topics within his own laboratory and also cancer epigenetics. He's an internationally recognized scientist, and we're very, very lucky to have him as both an individual, mentor, and peer. Secondly, we've got Dr. Adrian Sullivan, another colleague of mine, also in the Adelaide Centre for Epigenetics and the Cancer Epigenetics Research Program at SAiGENCI. She's head of the Lab of Development in Epigenetics and is formerly a Rutherford Crick Fellow from the Francis Crick Institute in the UK. Her work is now directed towards understanding cancer drivers and epigenetic mechanisms, and she's currently investigating a major gene critically associated with breast cancer progression. And so I'd like to bring our first speaker up to the stage, Jose, and kick the ball off.
Hello, can you hear me? Yes. And there you are. Thanks, Luke, for such a nice introduction. I'm his boss, so what else can he say about me? So I will start with development, and I know that it's a cancer seminar or lecture, but it's so we understand where we are coming from. So, as you know, during development, we start with the oocyte that gets fertilized, give rise to a zygote, which is one cell. Then you get the blastocyst, which is the early embryo. That's what is implanted in a woman when it's doing IVF, for example. And then everything goes well, the embryo gets implanted, generates at that state a trilaminar embryo. Everything goes super well, you get a beautiful baby. If you are lucky like me, you get a twin sister. Of course, they don't come from the same zygote, but you get the idea of how this works. So the amazing thing about my twins, and all of you, and myself, everybody, is that we are all made of different cell types. And if you think for a second on this, you already know that you have skin cells, blood cells that make the hair, cells in your mouth. All your cells are super different, neurons, etc. But they all came from one cell, the zygote. So there is no doubt that the zygote has all the information to make every cell of the body. That's very clear, yes. Now, many years ago, the question is, what happened with this information during development? Do we still have it? Do we lose it, etc.? So John Gordon did an experiment in the 50s, late 50s, where he took the nucleus of a cell, of an adult cell, of a xenopus, which is a type of frog, and put it back into an egg without the nucleus. And with that, he was able to make again an entire organism, a nucleus. That's cloning, basically. But that experiment demonstrated two very important things. One is that the information is not lost during development, so every cell of your body has the capacity to make yourself again, which you don't think on this every day because you have different jobs. I think on this every day, yes. Like every cell of our body has the capacity to make an entire organism again. Now, the corollary of this is that there must be something else than the genetic information. Because if every cell of the body has exactly the same genes, the same information, how can it be that I have the cells of the skin and are so different from blood cells? So the reason is because the DNA is, the information is in DNA and is organized in these bodies of information that we call genes. And all of you have heard the word genes, genes, genes. So we have around 20,000 genes. So the genes are like, they are encoded in the DNA and end up making, when they compact, end up making the chromosome that is like that cross there. You may have seen a karyotype, for example. So that is not it's a cartoon, but actually that's real data. Chromosomes look like that when you put it into in a microscope. So all the information is there. Those 20,000 genes are there. Now, those 20,000 genes, as you can imagine, the reason that some cells are different to other cells is because those genes are information that can be open and ready to be read, or they are closed and they cannot be read. So depends which genes, yes, the genes are closed or open. So the 20,000 genes are not all open at a time. So the idea is that, for example, a skin cell, a blood cell, have some genes open, some genes closed, and a skin cell has other genes open and other genes closed. And that's the difference. So the difference is not that they have different genes. What makes them different is which genes are readable. The example that I always said is like a university, and imagine that every faculty has exactly the same library with absolutely every book. So if you are a biologist, you go to the faculty of biology, you go to the library, and the librarian only opens the books of biology. So you are there, you become a biologist, you don't have other option. You study, I don't know, you go to the school of law, you go to the library, you know which books are going to be open, just the books of law. And sadly, you become a lawyer. So that's how it is. The cells is the same. They become whatever gene is open or closed. And this is basically, you have some genes open, some genes closed. And by the way, I'm simplifying this, but not so oversimplifying, because we talk in the lab, we talk about the gene is open or the gene is closed. So the way that this works is there is these proteins called transcription factors. And these transcription factors, there are around 2,000 in the body, different 2,000, are the ones that manage which genes, which books will be closed and open. And it's like the librarian. And in order to do this, the way that this works is they recruit what is called the epigenetic machinery, which is basically chemical reactions, yes, that get, literally, if one of you is a chemist or remember chemistry from school, is methylation or dimethylation or acetylation. It's not fancy chemistry here, but these little modifications and the combination of those little modifications is what makes a gene close or open. Yes, and those transcription factors govern this. So what has to do this with cancer? So as I told you, this has to be a lot with what a cell is. So same information, but a cell can be a blood cell, or a cell can be a neuron, or can be a skin cell. So in cancer, what happens actually is you have a normal cell, and two things basically happen usually. One is the transcription factors get mutated. And that mutation brings a different or a epigenetic machinery and starts to activate and change how genes are open and closed. These genes change, and now when the genes change, what are open and which ones are closed, is that the cell becomes a cancer cell. Yes, so simple like that. Basically, you have a program that basically is misreading the genes and starting opening some genes that shouldn't be open and closing some genes that shouldn't be closed. And if you think about this, it's not the mutation per se or the classic like BRCA1, BRCA2, or you have heard maybe about P53 mutations, AKT, etc. It's what they do. And in order for a cell to acquire, for example, the capacity to invade, which is what a tumor does when it invades, it needs to acquire other functions. So what happens is upregulates, yes, activates the genes that are involved in cell mobility. For example, some blood cells have that capacity. So what happens is tumors on the liver activate these genes, these books, and now the liver cells have the capacity to go and start moving when they shouldn't be moving. Yes, so it's as simple as that. So in the epigenetic program, we have, as Luke was saying, different groups, and all of us, we are trying to understand and control that apparatus. So in my case, I will talk more about that, what I do, so I will skip it. Adrian also will talk about it. Both of us, we work in transcription factors in trying to understand how those transcription factors, those librarians, control the cell identity. And Luciano works in a more holistic things. He tries to understand the entire cell. So how all the cells he measures at a time, and what, like in one measurement, the 20,000 genes, they are open or closed. And in that, he does this at the single cell state. So in a tumor that is, we know that you have heard that tumors are not the same, they are all different. He can say this part of the tumor is different from this part and different from this part. That's why he specialized. And Xizhang, she specialized in trying to target the epigenetic machinery. Why not? If the epigenetic machinery is the one changing which genes are open or closed, let's change the epigenetic machinery with a drug, and now there is no changes. So that's what she's trying to do. And then Luke, he's trying to target the interface between how the transcription factors, the chromatin, the DNA, and the epigenetic machinery are all working. And you can ask him more questions outside. So we are all basically trying to target different parts of this mechanism. So I work in what is called cellular reprogramming. That is basically, if every cell contains the same information, and it's a matter of which information is being readable, displayed, if we can control that, we can control cell identity. And indeed, what we do is we use transcription factors, we just put them in the cells, and the transcription factors start changing which genes are open or closed, And by doing that, the cell change and the information that is being displayed, and the cell change from, in this case, a fibroblast to, for example, a reprogrammed cell. And this is what we do every day. And it's not just like a blood cell, we can do every cell. No kidding, we can grab skin cells and make cells of your brain, neurons, we can make cells of the blood, we can make cells of the, in this case, kidney, that are cells of the placenta, so the organ that you left behind when you were born, yes, or and cells of the heart that were beating there. So this is something that we do all the time. And as you can imagine, the potential of this technology is amazing. You can replace every organ. And that's exactly what we are working on, yes, regenerative medicine. In trying to replace cell types that for some reason you lose them during in your life. So imagine, I always said this, if I can convert, well, I cannot do anything. The people in my lab do this. Yes, I haven't forgot even how to pipette now. But if the people in my lab, which are sitting there, yes, they can convert a skin cell into a brain cell, of course we can convert a cancer cell into a normal cell. It's the same process. And the idea is that, if a cancer cell is a cell that became raw, become different from what it needs to be, why we cannot reprogram it back into used to be, or at least identify which are those transcription factors that are driving that crazy program and just basically get rid of it. So in order to do that, we need to identify those transcription factors. I told you there is 2,000 transcription factors and they work in combinations. So how we find those transcription factors that are so important for that cancer cell? So many years ago, with a group of mathematicians, we created an algorithm that, this is with Owen and Julian, we created an algorithm that basically changed the 1,500 to 2,000 transcription factors into 8. So we can use that algorithm for each cell to say these are the most important 8 transcription factors. And the combination, when you work in combinations, for example, 1,500 transcription factors in combinations of 3 is around 3 billion combinations. So imagine when you work. So being able to pin the 8 that you need is super important. So by using this equation, with a group of prostate cancer researchers, we say, okay, let's test this idea. So I joined forces with Renee Taylor and Natalie Lister and David Cook, and we decided to go and try to see, can we use reprogramming approaches, basically how we control the genome, how the gene is expressed for prostate cancer. So as a proof of concept, we took a tumor from a patient that had died of prostate cancer. This was a metastasis in the dura madre in the head, and that tumor was is called a PDX. It's grown in a mouse, so we can now do experiments. And what we did is we did RNA sequencing, that is just a technique to look at the 20,000 genes, which ones are open, which ones are closed. We apply Mogrify, this equation, and it told us what are the top transcription factors that are controlling that cell identity. So we said, okay, if these transcription factors are controlling this cell identity, if we now shut them down, something should happen. So in order to do that, we use, at that time, a new technology. Now all of you know about this technology. It's called RNA, yes? So we use RNA, but instead of an RNA that gives you an epitope, like the vaccine, this is an RNA that takes away a protein. So using RNA anti-target, we basically put this RNA anti-target, our specific targets, those transcription factors that we said that are controlling that identity. And the idea is that you put it, and if it's working, the cells that become green, because the RNA is green, the cells that become green will disappear. So we found 2 targets, yes, and these are the cells with the control one. You can see they all become green, nobody dies so far. And when you hit the target, all the cells disappear. These are 2 real targets, L2 and L1 and MIBL2. And you can see that as soon as we hit it, 10 days later, there is almost no green cells. And actually, MIBL2 was also found by our director using another strategy, which is very good because basically using 2 strategies, we found a new target. So this is how we are using this type of technology. We identify the target, and then we can knock it out. So this is great, but as you know, every patient has different tumors, it's all different. And this is the good thing about this technology. What we did is, we went and we sequenced our more than 30 tumors from patients, and we look at all those targets. So basically each column is a different patient, and each row is a different potential target. And what we realized is that we were able to find common targets, so targets that we could knock out and kill all these tumors, and targets specifically for a subtype of prostate cancer, targets specifically for another type of prostate cancer, and then even patient-specific targets, which is like what we want, because are so specific that only those tumors will die, and you let the entire body not suffer. So that's how we are applying all this technology, and that's how we use epigenetics and the knowledge of epigenetics to target this. So in summary, there is a lot, but the thing that I would love that you remember from today is every cell of the body has the same genetic information. That's key, and it's magic, basically. You can do so many things with that. The epigenome controls that activity. By the way, epi means on top, yes? So on top of the genome. And then in cancer cells, many genes are incorrectly activated, yes? And due to these mutations and issues with the epigenome, yes? And the program is trying to use this information, yes, to target, to find solutions to this. And then never forget that basic research is super important. You don't see it at the beginning like, oh, how the basic research will cure this. But it's the only way. By basic research allow us to understand the mechanism, and by understanding those mechanisms, we can come with different solutions to different problems. So this is all the people that works in my lab. Yes, I have a lab in Adelaide, as Luke said, in SAiGENCI and in ACE, and I also have a small lab in Monash University. Of course, you don't do this work alone anymore. You need a lot of people with a different set of skills in the university, outside the university, and you need a lot of money. You need a lot of funding to do this, yes. So anyway, I would love to answer any question, and now or later. Later. Yes, later. Thank you.
Testing? Oh, yep. Okay. Thank you very much, everyone, for coming out tonight. As Luke mentioned, my name is Adrienne, and I've only just started my lab, actually, as a co-appointment between the Adelaide Centre for Epigenetics and the Cancer Epigenetics program in SAiGENCI. I am actually Adelaidean originally, but I've spent the last 6.5 years working as a postdoctoral researcher in London, in the UK, for a big biomedical research institute there. So in terms of my accent, I might sound slightly British now, or I could just be Adelaidean. I can't hear it anymore, so it's an open question. After that, I got the amazing offer to come back here and start up my own research group. And so that's why I'm here. During my postdoc time, I was actually not working in the cancer biology field. A bit similar to Jose, I was a developmental biologist. But they're not so unrelated. You sort of need knowledge of one to properly understand the other. So although my lab is still not been around for even a year, what I'm hoping to present to you today is how I'm taking that knowledge and that experience and creating an approach to discover new things about cancer biology. So if we zoom way out, big picture thinking, I am interested in a lot of the same questions as Jose, and in fact as a lot of researchers around the world. Why is a cell the kind of cell that it is? Why is it not a different kind of cell? how, when, and why would a cell change identity, and how can we control it? In terms of cancer, a cell changing identity is abnormal. It's a sign of misregulation. But actually, in development, this happens all the time, and not just during embryonic development either. As adults, we have little populations of immature cells called adult stem cells that are constantly changing into mature cell types in order to maintain or repair our tissues. And this process of an immature cell becoming a mature cell is known as differentiation. As Jose has already shown you, that involves a lot of changes at the molecular level in order for a cell to change identity during differentiation. Not only do you have that change in DNA packaging, that opening and closing, that then leads to different genes actually being expressed, producing products, and that leads to different changes in the physical and behavioural properties of the cell. So by that I mean things like cell shape, how fast it divides, invasion, and response to its environment, to signalling molecules and that sort of thing. So it's an extensive process, and it's not a switch. You don't go from one cell type to another instantaneously. Rather, it's a process, and the cell will occupy a continuous series of intermediate states while it's undergoing that process. The factors that drive this process as it goes along are related to how, my microphone has stopped. Thank you. All right, we'll try that again. Continuous series of states. This continuous series of states is known as the cell trajectory or lineage. And so the processes, the factors that drive that process are accordingly called lineage-defining transcription factors, represented here by a teal-coloured twisty. No one in the UK gets that joke. This process in development is extremely tightly controlled. So there's actually a lot to interrogate and understand at the molecular level in order to fully understand how this process works. And the way we study that is by making cells undergo this process in the lab. So here we have some immature cells. These are stem cells. I've made them red just so you can see where they are. But they're all the same kind of cell. They grow in a group like that. And I can actually force them to undergo differentiation by adding a particular factor to the media. I've also messed with their genomes so that when they do start undergoing differentiation, they will glow green. So here they go. This is a sped up movie, obviously, but actually this only takes course over 2 days. It's really fairly quick to start seeing things like shape changes and expression of these differentiation factors as they come on. If we look at more changes in the cell, we can see that actually there's different cell types here. We haven't gone directly from one to the other. We're getting what's called heterogeneity, a mix of different cell identities. And that's because this particular differentiation system is a model. It's a model of how different cell types arise in development. It's also a 2D model. More common are 3D models, where these aggregates of immature cells differentiate into their different identities using a lot of the same mechanisms as they would actually in a tissue in a person. These are called gastroloids for this early development model, but also organoids that can model different parts of the tissue of different organs. And they're incredibly useful to study these processes. In particular, something I am looking at with these models is how epigenetic remodeling is involved. So, as Jose has already told you, DNA can be closed, inaccessible to the machinery of the cell, which means no gene expression. Epigenetic marks, on the other hand, can change this so that DNA is more open and permissive to gene expression. And you can actually change one to the other during the differentiation process. You need to, in fact, you need different genes on and off as the cell changes identity. And that's again mediated by our lineage-defining transcription factors. and the epigenetic machinery that they recruit. Epigenetic gene regulation is therefore quite a major factor that controls cell identity, and they are heritable, and by that I mean between cells. Organisms is a different question that I'm not here to talk about, but when a cell divides, the place of those marks will be conserved in what's called the daughter cells. Fun fact, cells are a little bit like ships or planes. They're all female in English, even when they're male. Epigenetic marks also do not change the DNA sequence. So although we have what is at this point decades of information from tumours from patients looking at all of the DNA changes, that doesn't contain any information about the epigenetic state of those tumours. And probably accordingly, for such a key mechanism, epigenetic reprogramming was not recognised as a major hallmark of cancer until about 2022. So, how can we understand how those epigenetic changes arise when a cell becomes cancerous? This is the model system that I want to employ. It involves culturing the immature cells and then differentiating them as they would in a tissue and reconstructing cancer-driving events. So for example, in our cultured stem cells, we can make them express foreign genes, transgenes. We can do genome editing using CRISPR-Cas9, if anyone's heard of that system, to introduce particular mutations. We can also co-culture them with other cells to create a signaling environment that they would experience in vivo. As we differentiate them, we can also change their environment. We can add signaling factors, for example, different hormone conditions. We can add chemicals, anti-cancer drugs. All of the above are things we can change to see how that affects the cell identities we get at the end, which we can analyse by looking at the full range of epigenetic marks and the full range of genes that are expressed at single cell resolution. And by doing this, we hope to understand how those differences in cancer cells arise, how that means different properties of cancerous cells, and alternatively, we hope to discover new ways to detect, prognose, and treat cancers on a much more individualised basis. That is the general approach. I'm now going to talk a little bit more specifically about a project that's being developed in my lab currently, which is understanding how different sorts of breast cancers arise. Breast cancer is a very variable cancer, even by cancer standards. There's really no one-size-fits-all approach to treating breast cancer. And it's not incredibly clear why that should be so. The mammary gland is one of those organs that has its own population of immature cells that then continuously differentiate into the mature cell types. And there are, when it comes to cell lineages, there's two main ones. There's luminal and basal. Although it does appear that under some circumstances, cells can jump between them, just to make everything more complicated. People in the audience may recognise those names. Luminal and basal are also subtypes of cancer, so-called because they somewhat resemble cells from that lineage, which implies that one way in which you get different cell types is based on the cell that the cancer arises from, although this is currently just correlative. When it comes to factors that drive these different lineages, there is one quite well-known one for luminal lineage, and that is one of our lineage-driving transcription factors known as GATA3. As a lineage-driving transcription factor, it can cause epigenetic changes in cells as they differentiate. Here is a little bit of real data of differentiating cells. The little peak regions you can see correspond to places on the genome that are accessible, so they are open and correlated with activity. You can see as our cells differentiate up to 72 hours, so 3 days, you've got these regions here which are initially silenced and closed but start to open up. If I take those same cells and create mutations that mean GATA3 stays off, those regions no longer become as accessible as the cells differentiate. And if I do the same thing with the very related factor GATA2, which is a little bit redundant, it can do some of GATA3's job, that remodeling now doesn't happen at all. So the cells differentiate, but those epigenetic changes do not occur. And that has consequences for gene regulation, specifically this gene. This gene should turn on when the cells differentiate, but in the mutant cells it stays off. So, people might have realized what I'm getting at. GATA3 is important for differentiation. GATA3 is an epigenetic remodeler. GATA3 is a proposed driver of breast cancer. In fact, GATA3 is mutated in 10 to 15% of all breast cancers, which is quite high for breast cancer. It's very variable. It's also more commonly seen in younger premenopausal patients. And these mutations do not turn off GATA3. You can see that in this little map of all mutations, they more commonly occur in the second half of the protein. And what that means is you normally at least make half a protein, and then there's chaos. In terms of the proteins that do get produced, this top one is the normal case, the protein and the different parts that do different jobs. But the mutations that are found in breast cancer produce a bunch of different proteins, some with very small changes, some that are just straight up missing this part which interacts with DNA, or maybe they have part of it, or maybe they just don't stop and they just keep making some protein for whatever reason. As a molecular biologist, those look incredibly different to me. But as someone without a molecular background, they're all classed as GATA3 mutant. And so you would have GATA3 mutant cancer. So something I'm hoping to address is when we need to look at these things with more detail. Are those mutations really going to produce similar outcomes in breast cancers? Or do we need to start addressing them based on the protein structure? Another critical part of that question is related to the fact that GATA3 drives the differentiation of the luminal cells. It's there creating those changes over time as the cell moves to a more mature state. So therefore, which cell experiences that first hit, that first mutational event, could be a less mature cell or a more mature cell. And you would expect that that would have a different outcome on the epigenetic marks that that cell has when it experiences that activity change, that mutation. So one of the projects in my lab is taking mammary organoids, introducing these different GATA3 mutations at different points in development, and then seeing how that affects the epigenetic profile, the gene expression profile of the cells that are produced. And by looking at those signatures in those cells, we want to match them to cancers in cancer databases and see how that might be related to the pathology of breast cancers in real patients. Using epigenetics for this is particularly powerful because those marks are inherited cell to cell. Epigenetics not only reflects the cell's current properties, but also its history, its developmental history as it moves through that lineage. So in summary, cell identity changes in normal development and in cancer are actually two sides of the same coin. They use the same mechanisms. It's just that sometimes those mechanisms are changed, should be off when they're on, should be on when they're off, to create those outcomes that can lead to cancer or progress cancer. The aim of my lab is to implement cutting-edge molecular techniques together with these cell biology culture methods of development to determine how different variables such as mutations in key lineage-defining factors, this particular cell of origin, both in lineage and in differentiation stage, and the developmental environment that the cell is in produces different cancer cell identities and how that relates to different pathologies. And the ultimate goal of all of this, of course, is to define these specific epigenetic signatures which we can then look for in patient tumours and read that information. So I just want to say thank you very much for the members of my fledgling lab who were courageous enough to join an up-and-coming lab, Kate Dredge and Ashley Geiger. Kate is doing a lot of the molecular biology work. Ashley is tackling organoid creation. Also, we've had some placement students, Anna and Sateri from the University of Adelaide, who have worked on cloning mutant GATA3. Of course, I want to thank my colleagues at the Adelaide Centre for Epigenetics and the SAiGENCI Cancer Epigenetics program, Jose, Luciano, Luke, Chi, and Lisa, in particular Jose, for being an excellent boss and mentor and helping me get started on this exciting new stage of my career. Thank you again.
And so with that, I'll ask you to give one more round of applause to both of our speakers for today. And I'll ask them both to come up to the front and field any questions that we have from the audience. We'd be delighted.
Jose, thank you very much, and Adrian, thank you very much for that presentation. And Jose, congratulations for describing epigenetics in a way that I think that made it a lot of sense to people who don't understand epigenetics. My question for you is, so you've identified potentially 8 really important transcription factors that are involved in controlling cancer cells. Do you need to turn all of them off? Do you need to only turn one of them off? Or is it a combination? And then how do we take what you've shown here, which looks quite simple, and then transition it into something that can be used for patients who've got cancer?
Hello. Excellent question. So, actually, when we started, we didn't know how many we will need to turn off. And what we knew, based on past work, is that usually cancer cells get what we call addicted to 1 or 2 transcription factors. They get addicted to that program. And what we found actually is that, so we predicted, let's say, for that prostate cancer, specific for that patient, yes, 8 transcription factors. We tested them the 8 individually, yes. We did it first individually because it was easy, yes, just to put one by one. And at least 4 of them just alone kill the tumor. So you don't need to hit several. Now, maybe if we hit several, we get even better results. But better is because we go from, let's say, 100% viability, so the cells are all alive, to in 10 days, you only get 20% alive. And maybe if we, and at that time, it becomes very difficult. Under 20%, it could be many things that the cells are not growing well. So, but maybe if we hit 2 at a time, basically faster, or every cell dies automatically. So we think that with 1 or 2 is usually, based on the literature and what is actually in practice, is good enough. And how do you take this into a patient? So there is many ways actually, and it sounds easy, and actually it's easy in a way. It's more difficult. It's easy to find the target nowadays. So we could do this for every patient. So I showed 30 samples that are real samples, yes, and we found all those targets, and we can theoretically predict, let's say, for each of those tumors, 8 possible targets. In 1 month, we could test them empirically and say, these 2, go for it, yes? Now, how you translate that into a real therapy, there is different ways. One is you try to find a drug that already does exactly what shutting down that transcription factor do. We have done that. I was going to show a slide where we basically grab, and so we shut down that transcription factor and we go and measure all the genes again, and then with whatever happened, we check that information and we put it into an algorithm that tell you, oh, this drug does something very similar. And maybe the drug is used for completely something different. And we only look for drugs that have been already approved for using humans in different other things. And sometimes you find drugs that are very similar. And we have tested this, and it works. So that will be the easiest way, yes. And this is, for example, Nora, that is a bioinformatician here working with me, is the type of work that she does. And then we could go to a doctor, a physician, and that way in our team we have that physician. And depends the case, they could try to repurpose the drug to that. So that will be an ideal scenario where everything got lucky. So basically we need to get a sample from the patient, measure it very fast, find the target, said, oh, this could work, and that. The other is, now there is other technologies that we are trying to develop in SAiGENCI. There is two types. One is using these RNAs that I showed today. This guy. Don't worry, we are always prepared. So the other way is with RNAs. And 5 years ago, when we were starting doing those experiments, it was like, if I said, oh, we are going to use RNA, half of the audience will say, yeah, sure, whatever. But now, all of us have an RNA inside us when we got the vaccines. So the technology has come so much, it's so good now, that it's actually a reality. Yes, we could use it. That's one. The other is something that SAiGENCI is invested very hard on that, which the name is PROTACs, doesn't matter. It's a specific drugs designed to target transcription factors and kill them. And as the vision of our director was, if you guys are having all these targets, we need a way to actually then do something with them, yes, in the practice. So that's why we are developing this basically program also inside SAiGENCI. So yes, so it's not like this, but it can be done. Otherwise, we will not be doing this job. I will be starting something much more different, honestly. I truly believe that if we find those targets, we can do something with them. And I think that I can talk for all of us. Like, the best thing that could happen to our life, in our life, is saying, that patient, I got the tumor, I found the target, and we basically cure him or her. Yeah, that's the way. So yeah, it's possible. It needs time and investment, but it's possible. Thank you.
Thank you. I am not a molecular biologist, and this is a big picture question. I come from the theoretical physics background. It is actually about a member of my family who died young of breast cancer, and I was particularly fascinated by what Professor Sullivan had to say with epigenetics. I mean, when an insurance company, when someone applies to an insurance company for a life policy or TBD, one of the questions they ask is, have you had close members of family who've had cancer, particularly with women, breast cancer? It counts a lot against you. And yet, from what you're showing us today, is not 100% just hereditary. It could be environmental and emotional factors. Your comment, please.
Yes, thank you for sharing. I'm sorry for your loss. Definitely, it is not just hereditary. GATA3 in particular is not inherited at all, because it's involved in early embryonic development, which is how I came to work on it. So if you're inheriting mutations in GATA3, you have much bigger problems than breast cancer. You will probably not even make it to embryo stage. So all of those examples are things that have arisen in adult life. And if they arose in, for example, that mammary stem cell population, they could be around for quite a while until that stem cell is needed to differentiate, which is why there's often women who realise that they become diagnosed with breast cancer during pregnancy or after birth, because there's been a lot of growth in breasts, so that whole organ has remodeled during that period of time. There's been a lot of cell expansion, a lot of cell differentiation, and as a result, there may have acquired mutations, or mutations in stem cells have made themselves known. So absolutely, it's not just a question of your genetics. There are a lot of other factors that we do not understand properly. And I think the best way, preventative screening, of course, has come a very long way, and we're trying to get as much out of that as we can. But because there are so many different factors that affect cancers and give rise to cancers, ultimately, I think in terms of the treatment side of things, what we'd be looking at is tailoring a treatment to the specific kind of cancer that's arisen. With my non-medical background, that's as much as I can say on that, but it's definitely not a simple question with a simple answer.
Hi, I was really interested about the, I suppose, embryonic and transcription factors as a potential therapeutic target. I suppose my question is, what are the broader or widespread ramifications for potentially using transcription factors as a target or a therapeutic target? Are there broader side effects within the organism when you're affecting something that is kind of having such a major role at a cellular level? Is there issues there with those adult stem cell populations being affected by that transcription factor?
I think off-target effects is the question here. Yeah. Maybe.
So yeah, it's a great question, and that happens with any actually target in the cell, because the cells, the tumor cells, hijack the normal machinery. Yes, and that's why you see the secondary effects on all the chemotherapies. Yes. So it's a great question. So of course you couldn't target any transcription factor. Yes, you will need to find a transcription. There is 3 ways to see this. One is you need to find a transcription factor that is only happening there, for example. So sometimes they activate transcription factors that are only present during early development. So as an adult, it's not active. So classic is nano, is the name of the factor. It only is active on you in the first, 20 days of life, mostly, and then shuts down. But some tumors say, hey, this transcription factor is great, boom, up, and that's how it goes. So if you find one of those, it's a good way to go. The other is, as we were saying, they don't act alone. Transcription factors don't act alone. They act with other machinery, and that's what Luke tries to do. Instead of going for the transcription factor, you go for the combination, the specific combination that happens in the tumor cells between the transcription factor and the machinery. That even if the transcription factor is present in other cells, it's not using the same machinery, you use it only in the cancer cell, so you can activate that. And then the third option is what, this is common to any drug treatment, you can try to deliver it uniquely to the tumor. So in that. And then the other thing is, as I was saying, tumors usually become addicted to certain transcription factors. So even if it affects every cell in the body, affects more the tumors. So, and this is again across many drugs, you get the major effect, the maximum effect in the cancer cell, but a little bit in the other cells. So as far as is tolerable, you can do it. But it's an excellent question, no doubt about it. Whoever explained you development and transcription factor was really good.
Thank you for being here today, and our question are, what are the ethical considerations of implementing these research method in living patients?
Yeah, so I'm not a physician, yes, so I probably shouldn't be answering this question, and Chris could. And so this applies to any treatment, yes. There is nothing in particular with the transcription factor treatment per se that is different from other treatments in regards to the ethics. In order to do it, you will need to follow ethical guidelines, yes, that are dictated by the law that we have, by internal ethical guidelines that we have in the universities, in the hospital, etc. So you will need to follow all these guidelines and be sure that you are doing this in an ethical way. And you go through all this process of different clinical trials to know that you are trying to, the first thing, to not harm, yes, to do well. So that's the first thing that you try to do, not to harm. So that's why the first clinical trial, yes, phase 1 is, are we doing something wrong? If not, you go the next step. And that answers a little bit the question that I was asked first. Things take time because you need to be ethical conscious that you are doing the right thing and not doing harm. So that's, I think, my best answer. Chris, any?
Yeah. I will chime in super quickly to say, ooh, maybe I'll stand further away. To say that there's a lot of steps between what works in cells and applying anything to people. But the system of organoids has actually given us a new tool in that arsenal by first understanding how any of these treatments would work on human cells that are not in humans, that are in our dish. And then generally you then move to animal models, which are not human, but are a complete organism. Again, working ethically within the guidelines to minimise the use of those where possible. It's a very long road to get to humans, and you need to prove that you are operating as safely as possible, producing a good outcome for the patients.
Thank you for your talks. Have any of the transition factors been used on the BRCA gene at all?
The short answer is not really, no. BRCA is one of those situations where we're not talking about an epigenetic misregulation. It very much is a mutation with the associated outcomes with that mutation. The restoring activity of BRCA would be an entirely different question, unfortunately. And I could speculate, but it might be better if I just say that it's a different kind of issue.
Thank you. Just had a question about the GATA3 mutations and the different isoforms that you were looking at. Have you done any correlative studies looking at whether those isoforms correlate with patient response to treatments, like better or poor responders?
Yes, thank you for the question. I'm doing that right now. We're working with some a medical, what's the official name, Chris? Registrar, thank you, at the Royal Adelaide Hospital to dive into the cancer databases that we have access to at SAiGENCI. We're pulling out all the data from breast cancer patients. I'm going through and classifying the types of mutation in GATA3, and then she's going to look through to see if there's any evidence of differential response, rates of relapse, all of that sort of thing. It could be that all of those mutations do produce similar outcomes in patients, but to me, the next question is then why? What is the conserved mechanism between all of those different proteins that means it drives a cancer? Because if we can answer that question, that narrows the focus on what we can target in terms of therapeutics. I still think, though, that we're going to see differences in how they act and what that means for outcomes with the patient. So please stay tuned.
I think we'll have one last question and then draw it there for a close for the purpose of time constraints.
Firstly, thank you very much for a very interesting presentation. I'm an engineer and don't know too much about biology, but that all seemed to make sense to me, so well done. Your focus is directing at cancer cells, and as I think I've understood it, a cancer cell is simply a normal cell which has been given the wrong information, or the wrong pages in the book have been opened, so to speak, and it's not acting as the cell should be, so by shutting and opening the right pages you can get it to go back again. There are, and the cancer cell is considered a rogue cell in the system. There are other cells that I can think of, such as, say, scar tissue, which must be a living cell because it always repeats itself. From your explanation, it has to be a normal cell which has just been told to be a scar tissue. Is your understanding and what you're developing then could possibly turn scar tissue into normal tissue again? Is that the sort of level that we would function at?
Exactly. Yes. Done. We finished. No. Exactly. Yes. Yes. So that's the idea. And actually, reprogramming is now we are using it for cancer, but it's used for all what you said and more. So there is people trying to actually change the scar tissue and the cells that form the scar to normal cells or other types of cells. Of course, there is things that once created, they stay there and you will need to remove it in another way. Because the scar is not just the cell, it's all the things that cell has produced during the years and generates like a matrix of long proteins that stay there. So you will need to remove it. But completely in what happened. And indeed, this happened naturally because, for example, the liver, yes, where sometimes scars, and you know how the physician may have tell you, your GP have tell you, look, stop drinking for 1 month, give time to the liver to recover, to regenerate. Basically, that's exactly what is happening. The liver has the capacity is plastic and has the capacity to regenerate, so sometimes goes to the wrong place, and if you give the time and you stop giving some of the signaling that is making them becoming more of this scarring type, and fibrotic type, that's what we call it, it goes back into a more normal. And this you can do it for everything. And it's applied to, as you will imagine, to many type of disease in not only scarring, but for example, you can try to recover. So I have a student, for example, for kidney disease, where you lose one of those cells, it's called the podocyte. He's trying to make podocytes again. And the idea is you will do it in vivo. You will put an injection, change the cells that are around the kidney for the ones that you need, and you have them. Or for example, for Parkinson's disease, where one of the classic neurons is called a dopaminergic neuron is dying, you are losing them, well, why not to change the other cells that are around into a dopaminergic neuron? And actually what I'm just saying, there is some clinical trials on this. So yes, it's exactly what you are saying. But in cancer, something that happens, which is very crazy, is there is these long mutations. So it's not just like a piece of, an instruction in the page is crossed or not going there. That is what is called a translocation. So for example, you get half of the book from one book, and the other half comes from another book. So imagine the mix that you do when you mix something like that. So you get, for example, the first instruction is you need to be active all the time, and the other is you are going to control every tumor gene. And that happens actually, and you create like a super protein that is this. It's like you have the first part is, I don't know, Robin Hood, and the second part is Mary Poppins, and you jump from one to another one, and sometimes Robin Hood is taking care of kids, yes, or whatever. You know what I mean? It can be super dangerous the way that those put, but otherwise it's just information at the end of the day. Like you describe it perfectly, yeah.
So with that, I mean, it's a very, very difficult thing to take a very molecular concept like epigenetics and try and make it accessible to the public audience. So I would ask you to thank our speakers once more for dinner tonight. And we'll draw it to a close. I'll remind you that there are some refreshments on the way out, if you'd like, and otherwise feel free to come up to the stage afterwards if you have any more questions for myself or the speakers. And thank you.